Hello and welcome to the first video for the Higher Human Biology course on the first topic of Unit 1, which is division and differentiation in human cells. So the first thing we need to establish in this topic is the two main types of cells that we have in the human body and a little bit about how they divide and their various features and characteristics. So we'll start off first of all with the somatic cell. So the somatic cell is any cell in the body other than the cells involved in reproduction. So that'll be your liver cells, your brain cells, your muscle cells, all of the cells that make up your body other than those involved in reproduction. And we'll talk about those cells in reproduction in a moment or two. Okay, so try and get familiar with that word somatic cell. A somatic cell divides by the process mitosis, which you should all be familiar with from National 5, to form more somatic cells. During cell division, the nucleus of the somatic cell divides by mitosis to, and this is a phrase you'll be familiar with from that 5, maintain the diploid chromosome number. Okay, and that is a phrase it's going to be well worth you getting familiar with. Okay, maintains the diploid chromosome number, which essentially means it keeps the same number of chromosomes in the daughter cells that we find in the original cells. Okay, in humans, because we're obviously focusing primarily on humans in this course, it's going to be 46 chromosomes, but those 46 chromosomes are arranged in 23 pairs. Okay, and we can see from this diagram at the bottom here, that arrangement, okay, we get 23 pairs. And we call them homologous chromosomes because if you look at each pairing, you'll see that each chromosome looks very similar to the other one in the pairing. Okay, but then if you look at the pairs compared to other pairs, you'll see they are different from each other. And that's because you inherit one of each pair from your mother and one of each pair from your father, okay? So we say diploid cells have 23 pairs of homologous chromosomes. That's 46 individual chromosomes altogether. And you'll notice that last set of chromosomes there, which are either XX or XY, depending on whether you are female or male by sex, okay? So, and of course, there are other variants than XX and XY, but that's beyond the scope of the course. So we're not going to go into too much detail there, but you can always do some extra reading and research on that if you wish. So the second set of cells that we need to know about are the germline cells. So the germline cells are the gametes, that's the sperm and ova, and the stem cells that divide to form the gametes. Now, a common mistake that people make here is when they're asked about germline cells, they usually remember the gametes, but they forget that the cells that divide to form the gametes are also germline cells. Okay, so any cell involved in reproduction, not just the reproductive cells. Now, germline cells divide by mitosis to produce more germline cells, okay, just in the way that we've learned about previously, or by a new process this year called meiosis, and that's to produce haploid gametes. Okay. Now, at higher, we don't have to go into the process of meiosis in too much detail, but we do need to know about a couple of the steps. So first of all, the nucleus of germline cells can divide by meiosis instead of mitosis. Okay, so when a germline cell is looking to create a sex cell, a sperm or ova, then it will divide by meiosis. Okay, it undergoes two separate divisions. The first set of divisions involve separating the homologous chromosomes. Okay, so they line up at the equator as homologous chromosomes, and those two in the pair get separated to opposite poles. Now, the second stage is one that we're a bit more familiar with, where we are going to be separating chromatids in a similar way to how we did that in National 5 and mitosis. Okay, so there's two divisions, firstly separating homologous chromosomes, and secondly, separating chromatids. And lastly, haploid gametes contain 23 single chromosomes. Now, our diploid cells had 23 pairs of chromosomes, or 23 homologous chromosomes, but the haploid gametes contain 23 single chromosomes. Okay, so just one from each pair compared to the diploid cell. So that's the difference between the two types of cells, the somatic cells and the germline cells that you need to know about. Now, shout out here to a brilliant YouTube channel called the Amoeba Sisters. If you haven't yet subscribed to them on YouTube, I would suggest going and finding them just now and doing that. They produce brilliantly animated videos on a wide variety of biology topics. Now, the one word of warning I would give is that they are set up to primarily serve an American market rather than a UK or and specifically a Scottish market. So if you're sitting a Scottish exam, it will mean that you have to be selective about the content in terms of what you learn from it. I think that what they're excellent for is like a good overview of topics and processes and to visualize it and see it in animated form. But sometimes, the details they go into are different from the details that we have to go into as part of the Scottish qualifications. So keep that in mind, but do go and watch some of the videos there. They are great to watch and they're a brilliant channel. So moving on, cellular differentiation. Now, cellular differentiation is the process by which a cell expresses certain genes to produce proteins characteristic for that type of cell. Okay, now I would suggest learning that entire definition. It's a very common question in the past paper questions where it asks you to define cellular differentiation. Okay, and so knowing that definition is going to get you marks in all likelihood in your exam. Okay, so it's the process by which a cell expresses certain genes to produce the proteins that are characteristic for that cell type. So, for example, a red blood cell, it will not express the gene for a nucleus because it does not have a nucleus. However, it will express the genes that will give it a biconcave shape. Okay, so it only expresses the genes that will produce proteins that are characteristic for that cell type. And that's going to vary depending on the specific cell type that the body is looking to differentiate into. Okay, and this allows the cell to carry out whatever specialized function it has. And there's a wide array on the diagram there on the screen that you can see. Now, all cells tend to be different in terms of their appearance, and usually when structure is different in cells, the function is also different, okay? So the way in which they are built, the way in which they are differentiated will have a determining factor on their function, okay? Stem cells are the wonders of biology. They are unspecialized somatic cells. Now, they can do two things. They can either divide and make copies of themselves, which is handy, or differentiate into specialized cells. Okay, so make more copies of themselves or become specialized cells. Okay, and both of those functions are important for stem cells because throughout the body, there are a few areas where we have stem cells and we want to maintain that stem cell number. So when some differentiate, others will have to divide to maintain that stock of stem cells that we have within the body. Now, the two types of stem cells that you guys need to know about for your exams are tissue or adult stem cells. Now, they're involved in the growth, repair, and renewal of the cells found in that particular tissue. Okay, so we call them multipotent. Okay, remember that word multipotent. And what that means is that they can make all of the cell types found in that particular tissue only. So, for example, cardiac stem cells, ones found in the heart, can only make the cells that are found in the heart. Similarly, blood stem cells can only make the cells found in the blood. Okay, and it gives you the example at the bottom there. Blood stem cells located in the bone marrow can give rise to red blood cells, platelets, phagocytes, and lymphocytes. And we know from National 5 those are two types of white blood cells. Okay, so the blood stem cells can only produce cells found in that particular tissue type. And that's an important distinction to remember because in a moment we're going to look at a different type of stem cell which doesn't have those limitations. So the cells in the very early embryo or embryonic stem cells can differentiate into all the cell types that make up the individual and therefore are pluripotent. Okay, so you could take stem cells from this embryo and they would potentially differentiate into any cell type from that individual. Okay, so they have a much wider range of cells that they can differentiate into compared to the tissue ones. That's because all the genes in embryonic stem cells can be switched on. Now, they won't all be switched on. Okay, that would be chaos if you switched on all of the genes, but they can switch on any of the genes. Not all of the genes, but any of the genes. Okay, so these cells can differentiate into any type of cell. Okay, they can switch on the genes that make it into a skin cell, they can switch on the genes that will make it into a liver cell, or they can switch on the genes that will make it into a neuron. Okay, depending on the cell type that's required, embryonic stem cells can produce any of those cell types. Now, again, shout out to the Amoeba Sisters. They have a good video on exactly this process, okay? I'm not going to play the video within a video, but I'm sharing the image so that you can go and find this video on YouTube and watch it. It will be well worth it. The animation alone is excellent, and they also describe things in good detail and give lots of nice examples in context. So do go and check out the Amoeba Sisters after you've watched this video. Okay, so the therapeutic uses of stem cells. What can we use them for in therapy and treatment and in helping people? Well, we use them in the repair of damaged or diseased organs or tissues. Okay, and we're going to look at a couple of examples of those. For example, the corneal repair that we can see in the first diagram there. Now, the cornea is the outer layer of the eye there, and when there are issues with that, they can actually remove that part, put in a graft which has been grown from from stem cells, and replace the cornea in that way. They can also regenerate damaged skin. So, for example, if you have burnt your skin with, fire or acid or something similar, normally that would form scar tissue. But if you put a skin graft on it, which has been grown from stem cells, then that will help to repair that damaged tissue. And there's a lot of exciting things potentially coming up in terms of the therapeutic uses for stem cells. I'm a type 1 diabetic myself, and I know that they're doing a lot of research into potentially using stem cells to regrow the islet cells in the pancreas that secrete insulin. That's very exciting for me personally, but there are so many different studies going on for a whole wide range of diseases and conditions that will involve the use of stem cells to potentially repair those issues. So that's a very exciting time to be in, and hopefully as young people, as you become adults, you will reap the benefits of all of that research. And on that topic, it's worth noting that stem cells from the embryo can self-renew, which basically means that they're able to make copies of themselves if we put them in the right conditions in the lab. And researchers have obviously got that down to a fine art, because when you've got a supply of stem cells, you want to maintain that supply of stem cells so you can further your study and research, and you can continue to look at ways in which they can be used for therapeutic uses, for example. Okay, now I talked about research a moment ago involving the use of stem cells being used as, for example, model cells to study how diseases develop, or been used for drug testing. And of course, the reason they do that is because ethically, it's, I guess, more acceptable to use cells in the lab and program them to be diseased and then test drugs on them than it would be to test drugs on human subjects who have the disease. And for a number of reasons, because a cell just has, there are less variables, there are less factors involved, less risks, if you like. Whereas if you are testing drugs on a full human, then humans are very complex and multifactorial. So while the drug might be effective on the specific area that you're targeting, it might have other side effects. So it's good to test these things on cells first so that we can study them a little bit more, understand them better before we then start using these drugs on humans, okay? And for example, if you want to study a degenerative disease, for example, you can program those model cells to replicate what happens in those diseases, and we can see exactly what's happening inside the cell in a lab in the way that you couldn't if that disease cell was inside a human, of course. The stem cell research provides not just the model cells, but also provides information on how cell processes, such as cell growth, okay, and learning about how cells grow is so important, especially for conditions like cancer, for example. Differentiation, which is essential for us to understand how specific cell types are made from stem cells and how they're able to specialize. And gene regulation, so how genes regulate or express and then become the various proteins that make up an organism, how genes work in coordination with other ones, when are they expressed, etc. Okay, so there's lots and lots of cell processes that we can learn more about as a result of stem cell research. As I say, it's a very exciting time to be involved in stem cell research. There's so much new stuff being discovered all the time. However, all that said, there are many ethical issues with using stem cells. The use of embryonic stem cells, as I've already mentioned, can offer effective treatments for disease and injury, and there are loads more in the pipeline for the likes of Alzheimer's, dementias, type 1 diabetes that I mentioned before. However, it should be noted that it does involve the destruction of embryos, okay, and that's obviously the drawback, okay, that's the downside of using the embryonic stem cells. So effective treatments, but it does destroy the embryo. Now, the last wee section on this topic deals with cancer cells. So we're going to look at the biology of cancer and how it forms here, okay, as opposed to talking about all the other factors surrounding this condition. So cancer cells divide excessively. Okay, they divide excessively. That means they divide at a faster rate than they normally would. Okay, our cells divide and they divide fine. And in a way, cancer cells are not too different from normal cells. The main thing is they are not responding to our normal regulatory signals. Okay, those are signals which will tell the cell, hold on, you're dividing a little bit quickly, so let's slow down a bit, okay? But the cancer cells ignore or do not respond to those regulatory signals and therefore begin to divide excessively. Now, the result of that is a mass of abnormal cells, which we call a tumour. And you're probably familiar with that word tumour, okay? It's a very scary word, but that is what it means. It means it's a mass of these abnormal cells that have divided excessively because they haven't responded to the original regulatory signals that a normal cell would respond to. Okay. Cells within the tumour can sometimes fail to attach to each other and unfortunately can then spread throughout the body where they may form secondary tumours. And you've probably heard, in general chat, maybe family members with people on TV or programs or films, you've heard them talking about cancer spreading. And that's what this means. The cells within the tumour fail to attach to each other. And when they spread throughout the body and find a new place to locate, they can form these secondary tumours. Okay, and that's cancer spreading throughout the body. Okay, folks. So I hope that lesson's been useful for you and that, we've covered everything that you need to in that first topic. And I'll see you all for the next lesson. Hi there, folks. Welcome to lesson 2 of the Higher Human Biology course. We're going on to topic 2 of unit 1. Topic 2 is the structure and replication of DNA. Now, the base sequence of DNA is what forms our genetic code. We learned a little bit about this in National 5 when we looked at the 4 bases A, T, G, and C. And we learned a little bit about how they were complementary and how the sequence of those bases then dictates which proteins are formed. Okay, so the base sequence of DNA forms our genetic code, as it's called. Now, DNA is quite a long complex molecule, and it's made up of lots of smaller units, smaller repeating units. Now, if you're in chemistry, you would call that a monomer. And when monomers are joined together, lots and lots of them, they form something called a polymer. So that's going to come in handy later on when we're talking about DNA replication, just thinking about it in that way. But for now, let's just think of DNA as being composed of small individual units, and each of these units is called a nucleotide. And we can see, following the arrow here from nucleotide, we can see what one of these units looks like. So we've got a phosphate molecule, we've got a deoxyribose sugar, and we've got a base, okay, a nucleotide base. Now, those 3 elements make up a nucleotide. And for your exam, you would need to be able to state and label all 3 of these elements of a nucleotide. Okay, so the phosphate, the deoxyribose sugar, and the base. And fun fact, the deoxyribose from the is the first part of DNA, so deoxyribonucleic acid. So the deoxyribose sugar is what gives it that part of the name. Okay. Now, if we look at several of molecules all joined together here and we zoom in, okay, so this is one nucleotide at the top. This is a second nucleotide here, a third nucleotide, and a fourth nucleotide. What you'll notice is when those units, those individual units are joined together, they form a long structure here that looks a little bit like a backbone, like a spine. Okay, this is the spine of the DNA. It's called the sugar phosphate backbone because we have a sugar joined to a phosphate, joined to a sugar, joined to a phosphate, etc. So a sugar phosphate backbone. Okay, so in a single strand of DNA, we'll have these repeating individual units called nucleotides, and they will be joined between the sugar and the phosphate to form a sugar phosphate backbone. Okay, so that's the basic structure of the individual units that make up our DNA. This is a lovely little GIF here from the Amoeba Sisters, and you can see I love their animation, and I've already recommended their YouTube channel. If you don't already subscribe to it, go and have a look. The animations and their videos are brilliant. They've actually got a DNA structure video as well, which is great. So have a watch of that when you get a chance. So on to something we already know a little bit about from National 5, which is complementary base pairing. So if we remember from National 5, adenine A goes with thymine T, and guanine G goes with cytosine C. And our little rhyme for remembering that complementary base pairing rule was apples in the tree and cars in the garage. A with T and C with G. If you haven't quite mastered that from National 5, it's worth going over this again just to make sure you've got it for higher. If we have a look at our two strands here, okay, on the left, we've got one strand, and on the right, we've got a second strand because we know that DNA is a double-stranded molecule. Okay, so everything on the left here is part of one strand, and we can see our nucleotide units there that are then joined to another nucleotide and then joined to another nucleotide. And we can see the sugar phosphate backbone all the way down the side here. Now, on this diagram, we have actually named the base in each of the molecules here on each strand. So thymine, we can see here, goes with adenine over here. Cytosine here goes with guanine here. Adenine here goes with thymine here. Okay. Now, an additional thing that we have to note higher is that those bases, those complementary bases, are joined together by little bonds called hydrogen bonds. Okay. So hydrogen bonds hold those bases together, and that is what holds the two DNA strands together. Okay. So the two DNA strands are held together by hydrogen bonds. Now, a couple of extra little details about the structure of DNA that we need to know about. At one end, we call it the 5' end of the DNA strand. And at the opposite end, we have a 3' end. Okay, and then in the opposite strand, we have a 5' end down here and a 3' end up here. Now, you might be looking at that and thinking, why are they not at the same end? Why is on one strand the 5 at the top and on the other strand the 5's at the bottom, or vice versa with the 3's? Why are they opposite in terms of the direction that they're going? Well, DNA has this funny little structural thing where it's actually anti-parallel, which means that the two strands run in opposite directions. Okay, so this strand on the left here moves all the way down here, and this strand on the right goes from the bottom all the way back up. Okay, that's the direction that they run in. So they're anti-parallel. They run in opposite directions. Okay, think of it a bit like a motorway where you've got one lane of traffic going in one direction, and then just next to it, you have another lane of cars which are going in the opposite direction. Okay, they are anti-parallel to each other. Okay, so the last wee thing that we need to know is how do we know which end is the 5' end and the 3' end of a strand? Without going into the details of this, because you don't really need to know it for your exam, the deoxyribose sugar is numbered. Okay, starting with number 1 here where it joins to the base, number 2 down here, number 3, number 4, and then number 5 here, okay? And that's why this is the 5' end, because the phosphate is attached to the 5th carbon here on the sugar, okay? You don't need to know the chemistry of that, but you just need to know that the 5' end is where we find the phosphate, okay? And the thing I always say to my students is try and remember 5 phosphate, 5 phosphate. Okay, 5 phosphate. If you can remember that, you remember the 5' end is where the phosphate is, and the 3' end is where the deoxyribose sugar is, okay? And that's the 3rd carbon there. If we count from the base, 1, 2, 3, that's the 3rd carbon, which is the 3' end, okay? So 5 phosphate, 3' deoxyribose sugar. All right, folks, so that's the anti-parallel arrangement of our double strand of DNA there. If we move on, we get a nice little GIF again from the Amoeba Sisters, and they do the counting carbons thing quite nicely there. You can see that it's very visual, and you can see how we count from 1 all the way through to 5, and we can then work out which end is the 5' and which end is the 3' end. Okay, feel free to pause this video if you want to look at this a bit longer. Again, Amoeba Sisters have got great videos. I always talk about them. They have a good one on DNA and DNA structure, which you can have a look at. Also, just for a bit of fun, if you're not stressed and studying last minute for one of your exams, you can take some time out and watch Mr. W's DNA Rap, which is a good bit of fun. he sings about the structure of DNA. And just to be clear, Mr. W is not me, it's another Mr. W. Okay, so on to the second part of this topic, which is about DNA replication. So now that we know about the structure of DNA, we need to learn about how it gets replicated, how we make more strands of DNA. Now, the reason that we need to make more strands of DNA is because when we are making new cells through cell division, we need each of those cells to have the same chromosome complement. Remember that phrase, maintains the diploid chromosome complement? So in order to do that, we have to make an entire copy of the DNA that's in the original cell. So prior to cell division, DNA gets replicated. Now, the name of the enzyme which is involved in the replication of DNA is called DNA polymerase. Now, we mentioned monomers and polymers at the start of this lesson. The monomer being the individual unit, the nucleotide, and the polymer being the long strand of these repeating units. Okay, so DNA polymerase is going to join nucleotides to make a new strand of DNA by joining lots of these repeating units together to form a polymer of DNA. So that's why the enzyme that does this is called DNA polymerase. It will create a polymer of DNA. And another little handy hint, if you are unsure what an enzyme might look like, if you're unsure, and another little hint, enzymes usually end in A's. Okay, so if you're unsure what a word means, if it ends in A's, it means it's probably an enzyme. Okay, that's a nice little hint for all of biology, in all honesty. So, how does this process of DNA replication work? Well, first of all, we have to unwind the two DNA strands so we can get access to each of the strands and copy each of the strands. So the way in which DNA is unwound is by breaking those hydrogen bonds that we learned about earlier that join those nucleotide bases. Okay, so the bond that bonds A with T and C with G, we have to break that bond. Okay, so when that is broken, the two strands will unwind as we see in the diagram here. Okay, strand A and strand B are become unwound. They're no longer joined together. Now, these strands, A and B, will form what are known as our template strands. Okay, they're a template there to be copied. Okay, and then that will be what forms our new strands of DNA, which is how we're able to maintain that diploid chromosome complement. But before DNA polymerase can get to work and start making this polymer of DNA, we need something in place first. And that thing is called a primer, which we can see in the diagram here. Now, a primer is a short strand of nucleotides, which binds to the 3' end. Okay, we can see the 3' end of the template strand. And that allows then DNA polymerase to move in just after the primer and start to replicate the original strand of DNA. Now, we know that we use complementary base pairing to do this. So the DNA polymerase will bring the nucleotide that is complementary to A, which is going to be the base T. And then it will bring the next nucleotide in, which is going to be complementary to G, and that will be C. And then it'll bring in the next nucleotide base, which is complementary to C, that will be G. And then it will bring in the next nucleotide base, which will be complementary to T, and that's going to be A. Okay? So on this strand, on strand B here, that replication will happen in a continuous fashion, okay? As the DNA is unwound and we get the template strand showing a nucleotide base, the complementary base to that will be added by DNA polymerase. Okay, and that DNA polymerase adds those nucleotides to the 3' end. Okay, so it starts at this end and it works its way up. Okay, it'll work its way upwards in this particular diagram. And this process will happen not just at one place of the DNA, but all the way along the DNA at several locations, because DNA is an extremely long molecule. 3 billion or so base pairs within a strand of DNA. So it's a very long molecule, and we need to be then replicating at several locations just to actually get this done in a timely manner. So the process happens at several locations all the way along the DNA molecule. Now, on the opposite side, the primer will actually have to be added to, because it's anti-parallel, to the other side of this template strand, okay? It won't be added down at the bottom here, it will be added up at the top. And because DNA replicates in one direction only, okay, and this strand is anti-parallel, that means the primer is going to replicate, is going to come in here, and DNA polymerase is going to replicate in the opposite direction, okay, down this way. And we'll see in a minute why that's a slight problem, but also how our cells are able to overcome that problem. So if we have a look here, on the right there, we've got a nice GIF from the amoeba sisters. We can see the two strands been unwound and separated there. Now, don't worry about some of the names of these enzymes. We don't need to know all of these, okay? But what you'll notice is that top strand is just being replicated continuously in one direction from left to right, whereas the strand at the bottom there is being replicated in small little fragments. Now, the reason for that is because the strands in DNA are anti-parallel, okay? And because DNA polymerase can only add nucleotides to the deoxyribose, the 3' end of the new DNA strand, which is forming, that means that it has to do one of the strands continuously, but the other strand has to be replicated in fragments. And we can see that on our diagram here, okay? Continuous replication for this strand, fragment replication for this strand over here. Okay, and the reason for that, and this is important because they do ask about this in the exam, is that DNA polymerase only adds the DNA nucleotides to the deoxyribose or the 3' end of the new DNA strand which is forming. Okay, so therefore it's going to be replicated in fragments. Now, I did say that although this is a bit of a problem because we've got all these little sections of DNA rather than one continuous strand, the way that our body overcomes that, our cells overcome that, is they use an enzyme known as ligase. Okay, and ligase will join together those fragments to form one continuous strand. Okay, so it goes between the little fragments and joins them together. Okay, and we can see that in both our diagrams here on the screen. All right, so one strand is replicated in continuous manner in one direction, and the other strand is replicated in fragments. Fab. And again, the amoeba sisters have got a DNA replication video for you to watch as well. Okay, the last wee bit of this topic talks about a process called PCR, which stands for polymerase chain reaction. Okay, we've said polymerase a lot in this topic. Okay, remember polymerase is a enzyme which makes polymers. Okay, in this case, polymers of DNA, okay, or of the units that make up DNA, which are nucleotides. Now, PCR uses similar principles to DNA replication, and it's a process that we can do in a lab out with the body. Now, the reason that we do PCR is because we are trying to amplify certain regions of DNA so that we can do further tests on them. Examples, real life examples of where this is used are, for example, if you find a fragment of DNA, a cell or two at a crime scene, for example, you can then take that fragment, you can put it into a machine called a thermal cycler in the lab, and you can make lots and lots of copies of that. And that's useful because then you can test those copies, you can compare them to other samples that they've got on record, and they can potentially find the person on their DNA database via this method. But how does it actually work? Well, if we have a look at the wee diagram here, we can see, like DNA replication, PCR requires primers to be in place before any replication can occur. So before we can amplify that region of DNA, primers need to be in place. And a reminder that primers are short strands of nucleotides, and they tend to be complementary to the specific target sequences at the ends of the region of DNA to be amplified. That's a bit of a mouthful, but basically what it means is if you want to amplify this region of DNA, the primers have to be complementary to this end here, okay, so it joins on here, and this end here, so that replication can begin and we can replicate the rest of that strand and the rest of that strand of DNA. Okay, and we can see that happening there. The free DNA nucleotides are being added to this strand all the way along here. Okay, but only after the primer is in place. Okay, so slightly different from DNA. With PCR, we have to say that the primers bind to their specific target sequences. Now, when we're amplifying this PCR-led DNA replication, every time that we go through one cycle of PCR, we are doubling the amount of DNA that we have, okay? We go in with one strand. By the end of the first cycle, we have 2 strands. By the end of the second cycle, we have 4 strands. By the end of the third cycle, we have 8, 4 cycles, 16, 5 cycles, 32, 6 cycles, 64, and 7 cycles, 1 to 8. And that can obviously go on for a long time as well. You can do lots of cycles, okay? But that number just doubles every time. Now, again, this can be asked about in the exam, which is why I'm bringing it up. They will say, if, say, you have 1,000 molecules of DNA at the start of the process, how many will there be after 5 cycles, okay? And all you do is you double 1,000 DNA molecules 5 times. So 1,000 to 2,000, 2,000 to 4,000, 4,000 to 8,000, 8,000 to 16,000, and 16,000 to 32,000. So anyway, you would just double it each time. It's similar to the questions you did in National 5, where it talked about like cell division, how many cells are made after 5 divisions. It's the same process, only obviously in a different context here. Okay, and then what will happen is the thermal cycle of the machine that's doing this will go through repeated cycles of heating and cooling the region, the target region of the DNA. And that will then create all these copies. That will double our number of DNA copies every single cycle. Okay, so let's have a look at what actually happens during PCR, because there are 3 steps that you need to know about for your exam. So PCR involves heating, cooling, and heating. That's the way I remember it in my head, is I remember heat, cool, heat. Okay, initially, we heat up the DNA strand to between 92 and 98 degrees. Why do we do that? Because that amount of heat is going to break the hydrogen bonds and separate the strands. Okay, that's why we go so high with the heat there. And we don't have, or we don't use an enzyme to break the bonds. Instead, we use heat to do it. Okay, so they go between 92 and 98 and separate the strands. That's step 1 there. And we can see that in our diagram. The 2 strands have become separated. We then cool it back down a little bit. And the reason we have to cool it down is because the next stage involves us bringing in primers that are going to bind to those target sequences in the DNA. And in order for primers to bind, the temperature has to be cool enough that the hydrogen bonds won't break, okay, that the hydrogen bonds can form. So therefore, we cool it back down to between 50 and 65 degrees, and that allows our primers here, you can see them, X over here and X over here, we allow them to bind to the target sequences. Now, that's an important last bit there. I'd say when students are answering this question, that's the bit they miss the most often from their answer. So remember that. It allows the primers to bind to target sequences. And then lastly, we want to replicate this, this section of DNA. Now, we know that when we have high temperatures, for enzymes, that's both a blessing and a curse, because initially, as you increase the temperature, the enzyme activity will increase. But if the temperature goes too high beyond the range of the working range of the enzyme, then it will start to denature. So what scientists have done very cleverly is they have found a type of DNA polymerase that is heat tolerant. Okay, and it means it can work at extremely high temperatures. So they get the benefit of both working at higher temperatures and increased enzyme activity, but also the enzyme is not denaturing as a result. Okay, so they heat the thermal cycle back up to between 70 and 80 degrees, and that will allow the heat-tolerant DNA polymerase, which, fun fact, comes from thermophilic bacteria in volcanic hot springs, and that then replicates the region of DNA, okay? So that heat-tolerant DNA polymerase will replicate the rest of that region of DNA, and we can see the finished product at the bottom here, where we have 2 DNA molecules formed when initially we just had 1, okay? So heat, cool, heat. We start at 92 to 98 to separate the strands. We cool to 50 to 65 to allow the primers to bind to target sequences. And then we heat again to between 70 and 80 because that is the optimal temperature to allow the heat-tolerant DNA polymerase to replicate the region of DNA. Okay, it would be a good idea to memorize both the temperatures and the function and maybe make a wee table like I have on my PowerPoint here for you when you're studying and revising this topic. Now, the last thing on this topic talks about the practical applications of PCR. Now, I mentioned one right at the start there where I was talking about them finding a fragment of DNA at a crime scene and being able to amplify it. So obviously solving crimes is one of the main uses of PCR, but it can also be used in other ways, like, for example, settling paternity suits. If we do not know who the father of a child is and they want to genetically test so that they can ensure they know who it is, then they would use PCR to help them with this. To diagnose genetic disorders as well, PCR is also used in that process. And the way that it works is, so for example, the question I've seen in the exam come up more often is about either identifying a criminal based on crime scene DNA or identifying the father if you have the mother and child. And the way that they do it is like the one on the right here. So this is actually taken from one of the SQA exam papers, and I've taken out the bulk of the question because I just want to talk you through what's happening here. So we've got the mum there, the woman S, and we've got two men who are the potential fathers, and we've got two children as well. So what we're going to do is we're going to work out which father is the father of each of the children. So each of these little strands here, each of these little lines represents genes which belong to the parent. And those lines represent the genes that belong to the child. Now, we know that we inherit our genetics from our mum or from our dad. Right? We inherit 50 from mum, 50 from dad. That means that each child must have genetics that are also present in their mum or genetics which are also present in their dad. So what we'll do is we'll go through each child and we'll then check which parents have got the same genes as those child, and we can then work out which one is the mum and which is the dad. So let's look at child X first of all. Now we go all the way down here to this particular line, okay? Now, if we go across, mum also has that line, okay? So that doesn't give us any clue about who the father of that child is, okay? That's genetics from the mum. We go down to the next line, we can see that man P and woman S have these genetics, okay? Now you might instantly go, it must be man P that's the father. But because mum also has it, it's possible the child has inherited it from mum and not from that man, and therefore the other man could potentially still be the father. We go down to this next line here, child X, and then if we go across, neither man nor the woman have this shared genetics here. What that means is that none of them are the parents, okay? Or that neither of these men are the fathers, okay? It's possible that'll be the mum and it's a different man who's the father, or that's not the mum and it's a different mum altogether, okay? But we can't tell from this diagram. We go all the way down here and we see, yes, there's some shared genetics there with man P, but again, and then some shared genetics with this woman here, okay? But the problem is that we've got this one here that neither parent has, okay? So that is the issue for child X. Now, for child Y, we look at this top one, shared by both S and P, okay? But then we look at the second one, that's shared by Q. So both men are still in the running at this stage. Next one, we can see is shared by man Q, but by no one else. So we're leaning towards man Q, I would say right now, as the father of child Y. We go to the next one, the mum has it, that's fine, that doesn't really help us. Go to the next one, mum has it as well. We go to the next one, mum has it as well. So that suggests to us that man Q is going to be the father of child Y, okay? Because they have shared genetics, which we don't find in any of the other parents involved here, okay? Or the potential parents, I should say, that are involved here. And it's exactly the same with the examples at the bottom here with the parents here and the two males, and also for the crime scene one here as well. Okay, so anyway, we'll go look at one of these again when we look at the past paper questions in a few moments' time. But that's the end of this lesson, folks, and I hope that's been useful for you. As always, leave some comments and let me know how you enjoyed the lesson. If there's anything you'd like me to cover, if there's anything that you found particularly useful, I would love to hear about it. Thanks for now, though. Hello, welcome to lesson 3 of the Human Higher Biology course, where we will be going through the topics in Unit 1 in order, and we're now at topic 3, which is called gene expression. So gene expression is a term that's used to describe when a section of the DNA, we'll remember this from Nat 5, a section of the DNA which codes for a protein becomes expressed, okay? And that means that, to put it in a very basic terms, the gene is being switched on, which means that it will produce the protein that it codes for. So the first thing really to say about genes and gene expression is this, that only a small fraction of the genes within a cell are going to be expressed. You have a copy of all the genes that all of your cells need. So you've got 25,000 or so of them in there. Now, you're not going to be expressing all of those genes either at any time, or in fact, some cells will not express some of those genes at all, ever. So only a fraction, tiny fraction of those 25,000 genes are actually ever going to be expressed at any one time. And usually, again, we remember this from Nat 5, but usually one gene will code for one protein. We'll look at where there's exceptions to that a bit later on in this topic, but yeah, the general idea would be that if you got a section of DNA, a gene, it would have the code on it to produce a particular protein. So only a few, very small fraction of those genes are ever expressed at any one time. So we're going to look at the process by which genes are expressed and proteins are produced. So the protein production part involves two main stages, which are transcription and translation. Again, it's a good idea for you to remember those and also to remember which one comes first. And there's a couple of little tips that I've got for remembering which one comes first. The first one is both words start with trans, but then the next letter in transcription is C, and the next letter in translation is L. And C comes before L in the alphabet, so transcription comes before translation because of alphabetical order. That's one way to remember which one comes first and which one comes second. The second way is to remember that, and so for example, you'll be listening to this YouTube video. Now, if you want to translate this video into a different language, you would need to take the transcript, first of all, from this video, and then put it into a translator to change the language. So think about it like that. You've got to transcript something first before you can then translate it. So transcription and translation, they are the two main stages involved in protein synthesis. Now, last year we learned about messenger RNA. In National 5, we learned about messenger RNA, which takes a complementary copy of the DNA from the nucleus to the ribosome. Now, we're going to learn a little bit more about mRNA again this year and higher, but we're also going to learn about two other types of RNA called tRNA and rRNA. And we'll talk about them more in a few moments' time. But let's just talk about RNA just more generally just now. So RNA is in some ways similar to DNA. However, it has three main distinct differences that we need to know about for the exam. The first of which is DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule. The nucleotides on RNA have a ribose sugar in them, whereas DNA has a deoxyribose sugar. And again, the names of DNA and RNA will give you a hint as to which sugar is in them. So if you ever forget which sugar's in DNA and which one's in RNA, just think RNA is ribose sugar, DNA is deoxyribose sugar. And then lastly, the bases are slightly different in RNA. In DNA, we have adenine, thymine, guanine, and cytosine, A, T, G, and C. But in RNA, we have adenine, uracil, cytosine, and guanine. So uracil replaces thiamine in the RNA. So that's the three changes you need to know about. This does get asked a lot. It's been in a number of past paper questions, and it's well worth remembering those three differences between DNA and RNA. And there's little tables all over the internet, as well as in this PowerPoint, where you can see the difference between the two, and I would suggest memorizing those. So, quick recap on that 5. We mentioned earlier on, messenger RNA carries a copy of the DNA code from the nucleus to the ribosome, okay? And this little funky homemade flowchart will show you what happens there, okay? And hopefully you remember this from National 5. One of the big differences, we talked about it already, but the RNA, the messenger RNA, which makes a complementary copy of the DNA, is not exactly a complementary copy because it replaces all the thymines in the RNA with uracil. So where there's a in the DNA, and it would normally be a thymine complementary if we were making another DNA strand, in RNA, that's uracil instead. So you do the same thing as you did last year when you were looking at complementary strands, but the difference this year is if it's an RNA strand, instead of A being complementary with T, A is complementary with U, uracil. So keep that in mind. And the next part mentions that mRNA is transcribed from the DNA in the nucleus, and we can see that at the very top there. The complementary copy of the DNA in the nucleus forms our mRNA, and then it's translated into proteins by the ribosomes, which are in the cytoplasm. And we can see our ribosome here, and you can see the ribosome at work there, joining these amino acids together to form a protein. So hopefully all of that's reasonably familiar to you. Now, we're going to use a couple of different words to describe the base sequence on RNA, and this is an important one to remember. So each triplet of bases, each 3 bases on the mRNA molecule is called a codon. So you can see from, again, my funky homemade diagrams there, we've got A, G, and C, that's a codon. We've got U, C, and C, also a codon. A, U, and G, also a codon. C, U, C, A, C, G, and U, G, A, all codons. Now, what you'll notice is each of those codons, those triplets of bases, codes for a specific amino acid. Now, good news for you is for your exam, you do not need to learn the names of the amino acids or what the codons code for, which you don't need to know the specific amino acid they code for. You just need to know that they do code for a specific amino acid. Now, the one exception to that is sometimes there is a question in the exam where they will give you a picture a little bit like this one at the bottom here, which is a codon wheel. Now, a codon wheel is a way to work out which amino acid goes with which codon quickly. So the way it usually works is we work from the inside out here. The first base of the codon, let's look at our first ones there, that's A, would be here. Then the second letter of the codon, G, would be over here. And the third letter of our codon here is C, and that would be right here. And then that matches up with the amino acid serine, okay, or SER for short. Now, what's interesting about codons is sometimes the same amino acid is coded for by a different codon. And an example is here where we see UC for a second codon there. If we go to the codon wheel, in fact, sorry, U isn't actually on this codon, but if we used a codon wheel that did have it on it, we would see that UCC also codes for serine. So sometimes the codons, sometimes two codons, will code for the same amino acid. We just keep that in mind. Sometimes, even though they are different sequences of bases there, they will still sometimes code for the same amino acid. So keep that in mind. And you see the other codons, they're coding for different amino acids. Now, this last one here is of interest, okay? And that is the codon UGA. And again, we don't actually have UGA on that. This was taken from a past paper where you didn't need those. But if you look at a proper codon wheel, you would see that UGA codes for what's called a stop codon. Now, you can imagine that the function of the stop codon is to stop coding, okay, to stop the translation of the new amino acid chain in the ribosome. But we'll talk more about stop codon shortly, okay? But just be aware that sometimes it codes for something which isn't an amino acid. And those can either be start or stop codons, which, as the name suggests, either start or stop this process. So each triplet of bases on an mRNA molecule is called a codon and codes for a specific amino acid. Let's talk about the types of RNA then, because we've mentioned messenger RNA already. Now we're going to talk about tRNA and rRNA. So transfer, or tRNA, folds due to complementary base pairing. And we can see in this nice diagram here that it folds into this almost like T shape. And I always say to my class, that's how to remember what tRNA looks like. If you are given a diagram and it says something like, identify which of these molecules is tRNA, look for the one that looks a bit like a T. If you draw a line straight down the middle and across there, almost like a T. And the reason it's got that T shape is because of this folding that happens. It folds into that shape due to, and you can see the little hydrogen bonds there that cause the complementary base pairing within the tRNA molecule. Now, what's the function of a tRNA molecule? Well, the name transfer suggests it's going to be transferring something. And its job is to carry a specific amino acid to the ribosome. And the key word there is specific amino acid. Not any old amino acid, but a specific one. And it has a little amino acid attachment site at the top here where it binds to those amino acids. But how do we know which amino acid to bring? Because it says specific. Well, the thing that will determine that is this little bit at the bottom of the tRNA, which is called an anticodon. And an anticodon is a little triplet of bases, say a triplet just like our codon, but the anticodon will be complementary to the sequence on our original codon on the mRNA. So codons and anticodons are complementary to each other. So that's one thing to remember. And that anticodon is what tells the tRNA which specific amino acid to bind to and to carry to the ribosome, okay? It's these codes on the mRNA and the tRNA, the codon and anticodon, that determine which amino acid is taken to the ribosome. So a tRNA molecule, as I've already said, has an anticodon at the bottom, and that's at one end. And then at the opposite end, we have the amino acid attachment site. So the last type of RNA that we need to learn about for higher is the rRNA, okay, which is hard to say really quickly. RNA, or ribosomal RNA, and if you say the full name, it gives you a big hint as to what it is. Ribosomal RNA or rRNA plus proteins together form the ribosome. And if we look at this little image at the bottom here, we've got some ribosomal RNA, and you can see we've got the ribosome, but we've also got, if we zoom in, lots of proteins surrounding that rRNA. So the protein and the rRNA make up the ribosome. So if you want to do is a word equation, you could say protein plus rRNA equals ribosome, something along those lines. And that's all really you need to know about the rRNA. The tRNA and mRNA play a much more prominent role and feature more in what we're learning here. And as always, the Amoeba sisters have given us a gift here to help us understand the rRNA, mRNA, and tRNA functions quite well. So again, you can go and find that. They've got a YouTube channel which is hugely popular, and you can go and look at if you want to find out how they talk about these different types of rRNA. And they also have a great video on the differences between DNA and RNA that I've gone over. They go over it in a little bit more detail, probably more detail than you need to know for your exam, but it's still a very useful summary video. So let's move on then to the actual process of synthesizing these proteins. So what actually happens? Well, stage one here involves a enzyme called RNA polymerase. Now, we already met an enzyme called DNA polymerase from the DNA replication topic. RNA polymerase has a similar role to DNA polymerase, but of course, because it's RNA polymerase, it will not be involved in the production of a DNA strand, but instead the production of an RNA strand. And we'll talk about how that happens in just a second. So keep that in mind. And you always must, when you are answering in the exam, state which type of polymerase you are referring to, because DNA and RNA polymerase obviously have different roles. And when you're given the answer polymerase, you must state which one you mean. So RNA polymerase, it moves along the DNA. We can see it moving along the DNA in this diagram, and it unwinds the double helix. Now, technically, there's another enzyme that does this, but you don't need to know it for the course, so we'll just pretend that it doesn't exist. So RNA polymerase moves along the DNA, unwinding the double helix and breaking the hydrogen bonds between the bases. So unwinds the double helix and breaks the hydrogen bond between the bases. The RNA polymerase will then synthesize now, we call the mRNA that's formed initially, we call that the primary transcript of mRNA. And the reason we do that is because it's primary, it's first. Okay? Primary means first. So it's the first transcript, the first copy of the DNA. So that primary transcript of mRNA is made up of RNA nucleotides, okay? So RNA nucleotides that are joined to it by using the complementary base pairing rule that we learned about earlier on. So RNA polymerase adds those RNA nucleotides by complementary base pairing, and eventually that forms a mRNA primary transcript. And just to give you a sort of visual of what that looks like, I've done a wee handmade version here where I've got a DNA sequence, just some random letters, and then this is what the mRNA primary transcript would look like if we used complementary base pairing with RNA nucleotides. And what you'll notice is G goes with C, no bother, just as it always has. But you'll notice the A from the DNA goes with U in the mRNA primary transcript. That T in the DNA still goes with A. And again, A goes with U, and T again still goes with A. So essentially, if you were just to do a DNA copy here, then you would have T's instead of the U's, and all you do to make it mRNA is replace those T's with U's. So that's how we make our primary transcript of mRNA. And if only it was as easy as that and we just left it there. But however, some of that mRNA primary transcript has parts in it which we don't need. So we have to undergo a process next that's known as RNA splicing to help us form something that's called a mature mRNA transcript. And as you can guess probably from the name, the mature mRNA transcript is what comes later at the end of this process. We start with the primary transcript, we end up with the mature transcript later. These parts that we're looking to change and remove from the primary transcript, they are called introns. And the introns of the primary transcript are non-coding regions, and they are removed. We do not need them to code for our proteins, so we remove them from the transcript. And what we do then is we get these other coding parts, which are known as exons, and we join them together to form the mature transcript. And we can see that happening in a diagram where this exon coding, they are retained and joined together, and that forms our mature transcript. Whereas the section known as the intron, which is non-coding, that one is removed. We take that one out because we do not need that one. And what's worth also noting is that the order of the exons is unchanged during splicing. So if the code in that exon was CUC, you can see that was at the start, so that still remains at the start. And the code in this exon was UGA, and that remains in that order as well. We've just simply removed that intron in the middle there so that we've got this final mature transcript. Okay, so that's the mature transcript of the mRNA. RNA splicing just takes a wee second to get your head around, but the key things to remember are exons removed, exons retained and joined together to form the mature transcript. Okay, so that's part one, transcription, done. That happens in the nucleus. Then once the mature transcript is ready, it will leave the nucleus and it will move to the ribosome. Now, why does it move to the ribosome? Well, ribosome is where translation will take place. That's the second part of this protein synthesis process. So the translation of that mRNA, that mature transcript, into a polypeptide, and we'll talk about what that is in a second, that all happens at the ribosome. So let's have a wee look at our diagram here just to understand what happens in translation. So translation begins with something called a start codon, which is found on the mRNA mature transcript. Okay, we can see it here in the diagram. And it ends at a stop codon, which again, we can see at the opposite end of the mRNA on this diagram here. So the start codon at the start, the stop codon at the end. And basically what the instructions on those two do, they tell the ribosome to start assembling amino acids or to stop assembling amino acids. Now, while this is happening, while the mRNA mature transcript is moving through the ribosome and is being read 3 bases at a time, i.e. one codon at a time, at the same time, the ribosome will be letting tRNA molecules arrive, which carry a specific amino acid. Now, again, we said earlier on, how do we know which specific amino acid the tRNA should be bringing here? Well, it depends on what the anticodon on the tRNA is, because that anticodon on the tRNA, we can see the 3 letters there at the bottom of these tRNA molecules, these anticodons, they have to be complementary to the codon that is on the mRNA mature transcript. Okay, and we can see codons here all the way along the mRNA. So by complementary base pairing, the right anticodon will bind to the complementary codon, and in turn, that brings the correct tRNA, which is carrying a specific amino acid, into the ribosome. And then the ribosome will take the amino acid that has arrived and it will start joining it into a long chain along with the other amino acids. Now, those amino acids are joined together by bonds that are named peptide bonds. Amino acids are joined by peptide bonds. And what that means is, once enough amino acids have come in and have been joined by peptide bonds, we get what's called a polypeptide chain. And it's polypeptide because there are many peptide bonds, okay, repeating peptide bonds. So that's a polypeptide chain. Now, at this point, once the tRNA has dropped off its specific amino acid, it then will leave the ribosome. It will go away and probably get another amino acid and then come back to another ribosome and drop off that amino acid when the codon matches its anticodon. So that is the process of translation. Key thing is start and stop codons at the start and the end. That tells the ribosome to start or stop. We have tRNAs, which have anticodons on them that are complementary to the codons on the mRNA, so we know where to put the tRNA. When the tRNA arrives, it brings a specific amino acid. The ribosome then takes that amino acid and assembles it into a long chain that's joined by peptide bonds. And eventually that polypeptide chain will be the finished result of this process, and the tRNA will then leave the ribosome. So I hope that kind of makes sense. It's a tricky process to get your head around this, but just looking over the diagram, maybe redrawing it yourself, maybe watching a couple of the videos that are available on the Amoeba Sisters do a great video on this. So maybe go and watch that and you'll see like the animated version of it, and that might help you with understanding it. And speaking of the Amoeba Sisters, we've got a great GIF here from them showing both stages, the transcription stage where we make that complementary copy of mRNA, and then the translation stage where we have the tRNAs coming in with their anticodons, binding to their codons in the mRNA and then releasing their specific amino acid, which then gets joined by peptide bonds. Okay, so lots and lots for us to take in there. And this is the video that we're talking about. on protein synthesis that the Amoeba Sisters have done, and it's great. So just search them up on YouTube, subscribe to them, and watch some of their videos. The last wee section of this topic talks about an alternative way of splicing our RNA. Now, we talked about RNA splicing as part of the transcription process that happened in the nucleus, where the primary transcript had its introns removed and then its exons joined together to form a mature transcript. Okay. The issue with doing that is as follows. There are many more proteins that the body needs to synthesize than the number of genes that we have. So that creates a problem because usually one gene results in the production of one protein. So how do we produce different proteins from one gene? Well, we use this process called alternative RNA splicing. Don't get the two mixed up. RNA splicing is where the exons that are joined together stay in the same order. But alternative RNA splicing does something a little bit different. It changes the order of the exons. Okay. So, for example, in normal RNA splicing, we would expect exon 1, 2, 3, and 4 to join together and be in the same order. But by alternating this, by doing an alternative version of splicing, we can have, for example, exon 1, 2, and 3 joining together. That will produce a different protein than 1, 2, 3, and 4 would have. Similarly, we can take exon 1, 2, and 4, and that will produce a different protein again from both of them, from both 1, 2, 3 and 1, 2, 3, 4. And you could basically get all of these different combinations of exons. So, you could do 1, 2, 3, 4, and then you could do 4, 3, 2, 1, then you could do 2, 3, 1, 4, and then you could do 2, 3, 4, 1. And the number of combinations is reasonably large. And that's why we can make different proteins from the one gene by alternating the order of those exons. And it means that we can have lots of different mature transcripts that are produced from the same primary transcript, depending on which exons are retained and the order that those exons are retained in as well. So depending on which exons are retained, we can get different mature transcripts from the same primary transcript. All right, so that's alternative RNA splicing. That's when we want different proteins or more than one protein from the same gene. And the last wee section here talks a little bit about how we actually make a protein. So we got to the end of translation and we had this long polypeptide chain. That's a chain of amino acids joined by peptide bonds. But in order to make it an actual functional protein, we have to do more to this polypeptide chain. So first of all, we fold the polypeptide chain and we give it a three-dimensional shape. You don't need to know much about the shapes at higher. That's an advanced higher topic. But at higher, we need to know that when it's folded into these shapes, those shapes are held together by hydrogen bonds and other interactions between the amino acids. They help to keep the shape, okay? Hydrogen bonds and other interactions. They give that 3D shape. And because of this, we can basically fold it so many different ways, we have a large variety of shapes. And if we have a large variety of shapes, usually in biology, a different shape or a different structure equals a different function. So by having a large variety of shapes, we have a large variety of functions. And the shape of the protein helps to determine that function. And there's a little example on screen here where we've got a picture of the molecule hemoglobin. This is a protein that is produced. Now, we know about hemoglobin from National 5 because it's part of our red blood cells, and its job is to bind to molecules of oxygen. Now, the hemoglobin is so called because it has 4 little parts of the protein that are called heme groups. And you can see those little green groups there, the heme groups on the hemoglobin. And it's actually 4 little protein units all joined together. You can see that from the red and blue distinct colours in each of the subunits. Now, that heme molecule binds to the oxygen, and then that's what allows it to attach to the red blood cell and be transported around the body. But that shape is specific to that function. So we wouldn't have other cells that have this same shape and therefore this same function. Proteins have a different shape dependent upon their function. And the last wee thing just to note on this topic is that the phenotype, that's the physical expression of the genes, that's the physical appearance of an organism and the proteins that they produce, that is determined by gene expression. So by which genes are expressed and therefore which proteins are produced. Okay, so your phenotype, physical appearance, the physical expression of the genes determined by gene expression, this process that we have just learned about. But it's also worth noting that as well as your phenotype being determined by your genetics, your genes, and which genes are expressed, it's also partially determined by environmental factors. And something like skin color is a good example of that. Your skin color will vary due to a protein called melanin. And that's both a genetic thing, a gene expression thing, but it's also partly your exposure to UV radiation from the sun as well. That will influence the production of melanin, that protein within your skin. And so, therefore, that will also influence phenotype. So, environmental factors can also influence phenotype too. Okay, folks. So I hope that was useful and that you got a lot out of that lesson. As usual, leave me any comments on what you enjoyed, what you would like to know more about, or if there's anything you're still unsure on. We'll see you for part 2 in a minute, where we do some of the past paper questions. Hi, folks, and welcome to lesson 4 of the Unit 1 in the Higher Human Biology course. Today, we're going to be looking at the topic of genes and proteins in health and disease, in other words, mutations. So let's kick off with the definition of a mutation at higher level. We learned about them being a random change in the genetic material at Nat5. Higher, there's just a slight alteration to that. We say mutations are changes in the DNA that can result in no protein or an altered protein being synthesized, okay? So either no protein or an altered protein being synthesized. And the reason for that is because when there are mutations or changes in the DNA, that will then resultingly change the codon, which means a different amino acid will be brought to the ribosome to be synthesized into a different protein, okay? So changes in the DNA mean a different codon, a different amino acid, and therefore it will change the protein. We'll either get no protein at all, or the protein will be altered in some way. And we'll talk about how that happens throughout this lesson. So we're going to be looking at different types of mutations and how they impact the production of proteins. So we're going to kick off with the single gene mutations first of all. Now, single gene mutations are ones which involve the alteration of a DNA nucleotide sequence. A base, nucleotide base, is altered in the sequence, and that can either have minimal impact or quite a large impact depending on the type of alteration that occurs there. So we're going to look at 3 different ones, and we're going to look at the varying impacts of each of them as well. Now, if we look on the screen here, we can see that we've got a normal DNA strand here, okay? GAG, TGC, ACT. And when we convert that into mRNA and we read the codon at the ribosome, these produce these different amino acids, okay? We're just calling them amino acid 1, 2, and 3 for simplicity. There are names for the amino acids, but you obviously don't need to know all of them for the exam. So I'm keeping it nice and simple here. Now, the first type of mutation that is a single gene mutation will be a substitution mutation here. Now, a substitution mutation is where you replace a nucleotide base with another within the sequence, okay? So in our original sequence of DNA here, we start off GAG. In our substituted mutation, which is right here, substitution being right here, we see that the A that was in this one has been replaced by this T here. So it's been substituted. Now, if we look at the rest of the sequence, all the other letters remain the same as they are in this original DNA at the very top here. Okay, so there's only that one nucleotide base that has changed. Now, the impact of that is that it will alter one amino acid. Okay, it alters the amino acid that is coded for by this codon, but it does not impact the subsequent amino acids. They remain the same as they were. Okay, so substitution, where we're replacing one nucleotide base with another, has the impact of changing one amino acid, one codon. Okay, that's well worth remembering. The second one we're going to look at is insertion. Insertion, as the name suggests, involves adding a nucleotide base to the sequence. So we look at the original DNA here. We've got GAG, it starts. Now, if we look down at this one, this goes GT. So we've got an extra T in here. Okay, this T has been inserted into the sequence. So above here, it was replacing the A, but here it's been inserted and the A and the G are still there. Okay, they've just been moved along. Okay, all of the letters, you can see all the way along, all of these bases have been moved along one place. Okay, if we compare them to the original DNA. Now, the impact of that is that not only do we change the amino acid that's coded for here by this first triplet of bases, we then change every subsequent amino acid that comes after as well. Okay, so that now codes for a different amino acid here, and this one also codes for a different amino acid here as well. Okay, so that's insertion right there. And the last one we're going to look at is deletion. Now, deletion, again, as the name suggests, involves removing a nucleotide base from the sequence. Okay, so this one here is going to be our deletion. And again, see if you can spot what's been deleted. And we can see from the original sequence that that A is no longer there because the original sequence started G, A, G, T. And on this sequence, we've just got the G, G, T. So the A has been removed, okay? So there has been a little removal of that A from in there, okay? Now, the impact of that is that all of the nucleotide bases have moved again, but this time they have moved in that direction, okay? Whereas they moved in the other direction before with the insertion, with the deletion, they're all moving one place in that direction. Now, again, what this means is not only does the codon and the amino acid change for this first triplet, the codon and therefore the amino acid for every subsequent one also changes. Now, if we compare the amino acid sequence that makes up the first protein here, 1, 2, 3, and we compare it with substitution, we can see only one of the amino acids is changed. When we look at insertion, we can see 1, 2, 3 of the amino acids are changed, i.e. all of them. And when we look at deletion, also 1, 2, 3 have been altered and produced a completely different protein here. Now, we call those insertions and deletions, we call them frame shifts, okay? We call them frame shift mutations. Now, the reason we do that is because what we are doing with these mutations is we are shifting the frame, we're shifting the sequence in some way, okay? In this one, we're going that way, and in this one, we're going that way, okay? So we've got our insertion here, shifting the frame to the right. We've got our deletion here, shifting the frame to the left. Now, an interesting story about how that name came about, the frame shift. In old movies, they used to have a film reel and it had individual frames, okay, individual little pictures on the reel. And because they spun the reel so fast, those individual frames or pictures looked like a sort of live action movie. Now, when they were editing those reels, what they used to do was literally cut out a frame, or if they wanted to add in something, they would add in a frame, okay? They would insert or delete frames within the reel. And the impact of that is, obviously, if you inserted a frame into the reel, then all of the scenes in the film shifted forward a few seconds. And similarly, if you removed a frame from the reel, all of the subsequent all the subsequent frames moved backwards in the reel and would have occurred at an earlier point in the film. Okay, and it's the exact same for our DNA sequence here. Okay, frame shifts cause all of the codons, and all of the amino acids after the mutation to be changed. Okay, so that's a frame shift mutation, and that involves both our insertions and our deletions that we've learned about. Okay, so we call them frame shift mutations. What is the impact of this? Well, as you can guess from the fact that it changes every codon and every amino acid afterwards, it has a major effect on the structure of the protein that's produced, okay? It'll be completely different, and it'll either be a different protein, it'll be a non-functional protein, or even it might even code for no protein at all, okay, depending on how severe that change is. So that's frame shift, which includes insertion and deletion. Now we're going to talk a little bit more about substitution mutations because we briefly mentioned them at the start about changing one nucleotide base, and that affects one amino acid, okay? Now, nucleotide substitutions are split into three main groups. We've got the missense ones, we've got the nonsense ones, and we've got the splice site mutations. Okay, and we're going to talk about how each of those three substitutions are different from each other. So a missense mutation, that's this one here, results in just one amino acid being changed for another. So in this case, the original sequence has amino acid 1. When a missense substitution happens, that replaces amino acid 1 with amino acid 4. And if we look at the rest of the sequence, we see no change in the amino acids there. Okay, so we've just directly changed one amino acid for another. Now, this has the impact of perhaps resulting in a non-functional protein, okay, one that doesn't carry out the function it is supposed to, or sometimes it can have little effect in the protein. just one amino acid might not change the protein significantly and it can still do its function. Or we even have this scenario where because our codons often we have more than one codon coding for the same amino acid, it means that even when we change the codon, sometimes it might code for the same amino acid. So it might not even cause a change here. But anyway, we'll either have a non-functional protein or have little effect on the protein with the missense ones. Now, for the nonsense ones, okay, and that's them here in the diagram, they result in a premature stop codon. It's well worth remembering that whole phrase. A premature stop codon, which is being produced. And what that does is it results in a shorter protein, okay? And what do we mean by that? Well, for example, if the G from the original strand here is replaced by a T, substituted with a T, that then codes for not another amino acid, but for a stop codon. And we know from the previous topic that a stop codon will instruct the ribosome to stop translation. So therefore, the protein will end here, okay, or the polypeptide chain will end here. And the result of that will be that we get a shortened protein. So it's the premature stop codon, which results in a shortened protein. That's what this substitute mutation does, the nonsense mutation, okay? You might think all of this just sounds like nonsense, to be honest, but these are the names that are given to these substitution mutations. Okay, and the last type of substitution mutation is called a splice site mutation. Now, those result in some introns being retained and some exons not being included within the mature transcript. So if we remember back to the previous topic, we looked at RNA splicing and we talked about removing the introns and retaining the exons. Now, if there's a mutation at a splice site, i.e. between an exon and an intron, okay, in these areas here, if there's a mutation in those areas, that will mean that the cell won't be able to work out what to remove and what to retain. And when we have no mutation, we normally get exon, joined together, intron removed. But with the mutation involved, sometimes we will retain an intron in the mature transcript, or we lose this exon here, okay? And that is not retained. And we need that exon, and we don't need that intron. So we're ending up with mature transcripts here, which are not going to code for the right protein, okay? They are not going to have the right codons to make the amino acids which make up the protein that we actually want. So splice site mutations result in some introns being retained or some exons not being included within our mature transcript. Okay, and on to the last wee bit of this topic where we talk about chromosome structure mutations. Now, chromosome mutations can involve changes to either the chromosome number. Okay, remember we have 46 chromosomes within the human cell normally, but there can be changes to that if there's a chromosome structure mutation. And also changes to the structure, which is what we're going to focus on in the next little section here. So there are 4 different changes to the chromosome structure that can occur. And one of them actually shares a name with our single gene mutations, which is deletion. Now, deletion, and remember this for all your definitions for chromosome, you have to say is where a section of a chromosome is removed, okay? Remember that bit, section of a chromosome. So deletion is where a section of a chromosome is removed. And if we look at the diagram over here, we can see this C gene here, okay, this C part of the chromosome. When we go over here, that has gone, it's been removed, okay, it's been deleted. So deletion, removing a section of a chromosome. The second one, duplication. This is where a section of the chromosome is added from its homologous partner, okay? Now we know from the first topic that all of our chromosomes are in homologous pairs. So in duplication, we are getting a section added from the homologous partner. So if we look at this example here again, we've got A, B, C, D, E, F, G, H. That's fine. That's all normal in that chromosome. But in the duplication error, the C and D from the homologous partner have been added to this chromosome, okay? If we looked at the homologous partner, we'd see it lacked the C and D because it's gone over to this chromosome here, okay? So it's added from its homologous partner. That's duplication, okay? And you can see visually there how that works, CD, but then CD again. And they've got that extra C and D from the homologous partner. The next one is inversion. And if you imagine, if you invert something, if you invert your hand or you invert a chair, whatever, you're turning it upside down, you're reversing it, okay? So this is where a section of a chromosome is reversed. And what we can see here is if we look at the normal sequence, A all the way through to H in the normal order, when an inversion mutation happens, we can see that the B, the C, and the D have in fact been inverted, haven't they? They have been inverted and the whole thing has swapped round. Okay, it's like it's been flipped upside down, if you can imagine it like that. And the last one is probably the trickiest one for people to get their head around is translocation. Okay, now trans as a prefix in biology we know means to move to or to cross over to, and translocation means to move to a new location. So this is where a section of the chromosome is added to a chromosome, but this time it's not its homologous partner. Duplication is the homologous partner. Translocation is not the homologous partner. So if we look at this, A, B, C, D, E, F, blah, 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 and then we've got different letters here to represent the genes on the other chromosome, we can see that the A and the B have moved. Okay? They were on the top chromosome and they've moved down here. And the I, J, and K were down here and they have now moved up to this one. So a section of the chromosome has been added to its partner that it's not, and it's not its homologous partner. How do we know it's not homologous? Because it's got different letters, different genes, different sections on this. It's at a different length as well. Okay, so this is not its homologous partner that it has added a section of chromosome to. Okay, so keep those four in mind and try and remember the difference between all of them. The names give you a hint, but you need to know these wee definitions as well. And note that they all start with a section of a chromosome. Okay, so you have to mention a section of a chromosome when you're describing the change in a chromosome mutation. Now, what does this mean? Well, it means that the substantial, and it is substantial changes, remember, we're not just changing a single nucleotide base, we are changing large sections of a chromosome. Okay, so we are changing a lot. Now, the substantial changes in the chromosome mutations often make them lethal. Okay, so they are more lethal than the single gene mutations that we've already been learning about. Okay, and again, just keep that in mind as you might be asked about that in the exam. So that's the end of the lesson. Hello, folks, and welcome to Lesson 5 of Unit 1 of the Higher Human Biology course. Today, we're going to be covering human genomics in this lesson. Now, this is one of the shortest of the topics in the course, so it won't take us as long as our other lessons. However, there are some important things here that we make sure that we learn just in case it obviously comes up in the exam. And we'll do a couple of exam questions at the end as usual. However, again, there are not many questions from the past papers because it's such a small topic. So we'll do our best. We'll go through them all and we'll obviously go over everything. But just keep that in mind. It's not the most assessed part of the course. But nevertheless, we should still treat it with equal effort and learn all the content just as well. So let's talk about sequencing DNA. So when we talk about human genomics, and there's a very, probably seems very old to you guys, but happened in my youth. There's a very old project called the Human Genome Project, and that's where they, between 1990 and 2003, so 13 years they spent on this, and they spent over $3 billion on it. And it was a cross-country effort. Actually, it's had the scientific collaboration that we don't quite see as much these days in the modern world. But all the major countries and so on combined together to work on this. And what they managed to do is they managed to sequence the entire 3 billion base pairs that we find within the human genome, which is quite a remarkable feat. And as I say, at the time, it took 13 years and $3 billion. What's interesting is, like almost all technology, both the cost and the time to do this has come down dramatically in recent years. And only just a couple of years ago, I got my own genome mapped, my own human genome mapped, and it cost, I think it was £90, so less than £100, and it came back within a couple of weeks. And it's you know, the speed at which technology goes and the ability, obviously, of like things like computers and other programs as well to help with these things mean that tasks which once took us 13 years and $3 billion are now taking us a couple of weeks and £100 from, you know, a local lab. So, but sequencing DNA, it was once thought of as a huge challenge, and obviously now is something which is very much achievable for us, and we're able to do quite quickly. So the genome of an organism, I've mentioned the word genome a few times, and so let's talk about what it is. The genome of an organism is its entire hereditary information encoded in the DNA. That's a bit of a mouthful, that first sentence. I'd still try and memorize it as it is for the exam, but what that means in sort of normal terms is the genome of an organism is all of the genetic information that you have inherited from your two parents, and that's encoded in our DNA. Okay, so hereditary information. Now, the genome is made up of both genes, as the name suggests, genome is made up of genes, but actually is made up of a lot more other sequences that do not code for proteins. So we know that genes are the codes for proteins. We know that from the gene expression topic, but only about 1 to 2%, I think it's about 1.5 to 2% of our genome is actually genes. The rest of it is other sequences which help in things like gene expression and so on, and have lots of other tasks. Rather than just being genes that make proteins. So that's worth remembering as well. The genome is made up of genes, but there are lots of other DNA sequences that do not code for genes. So we talked about sequencing DNA, and the Human Genome Project was able to sequence the entirety of our DNA. So what is genomic sequencing? Well, in genomic sequencing, the sequence of nucleotide bases can be determined for both individual genes and the entire genome. So, for example, they could do genomic sequencing on your DNA and, in theory, could give you the full list of the 3 billion base pairs that exist within your DNA in the correct order as well. And you can see a wee image here at the top. I mean, that's a tiny fraction of what the genome would look like, but you can see how basically unreadable that is to most humans, right? If you looked at that, you wouldn't know where to start. But fortunately, there are people with a trained eye who do know where to look, and they've obviously worked on this for a long time. But more and more recently, the use of computer programs have been used to help scientists to identify base sequences by looking for some of the sequences that are similar to the genes that we already know about. For example, if you're looking for a gene through a genome for a particular condition, an inherited condition, and you know that from other patients who have the condition, they have a certain sequence on their DNA, then you can look for that within the person you're testing. Now, for a human to do that, to read someone's 3 billion base pairs and to find a sequence, or even if even if you're only looking at a small section of the DNA, even still, it's going to be overwhelming. So what they do is they use computers to help them with that, and they can obviously do that much faster and usually with fewer errors and more accuracy than a human might do. Now, the use of computers doesn't just stop there. When we are comparing sequence data from, say, a human and a sample found at a crime scene, or perhaps we are looking at how related two people are to one another or something similar, we use both computer and statistical analyses, which collectively are known as bioinformatics. And there's a little image here that just shows you that crossover between biology, statistics, and computers there, which all merge together to form that field of bioinformatics. So yeah, when we're comparing sequence data, we have to use our biology knowledge, but we also have to use our statistical knowledge as well as our computer science ability as well. The use of computers and all of the programs and software and so on that are available now on computers. That is called bioinformatics, and that's a word that's very much worth remembering, bioinformatics. So we use computer programs to identify those base sequences, and in genomic sequencing, we can determine the entire sequence for both individual genes and the entire genomes of people. The genome itself is made up of genes, but also other DNA sequences that do not code for proteins. And our genome is the entire hereditary information found within our DNA. Okay, so hopefully that first bit all makes sense. I get most of this stuff is just it's just about learning it. There's not likely to be any great deep questions in the exam on this topic, or certainly there hasn't been in the past. Most of the questions are just a multiple choice question, and we'll look at a couple of those at the end just so you can see how you're applying this knowledge. So the second part of this topic talks about the uses of these genomes. When we sequence someone's genome, what can we actually use that information for? Well, an individual's genome can be analysed and it helps us to predict the likelihood of developing certain diseases. Because what they can do, as I said a moment ago, is they can look for known sequences. That's sequences they already know about that they can attribute to a particular disease. And if you have that sequence within your genome, then they know that you are likely to develop that disease. And we can use pharmacogenetics, which is the use of genome information in the choice of drugs. What they can do is they can use an individual's personal genome sequence, and they can use that to then select the most effective drugs at the most effective dosage to treat their particular disease. And we call this personalized medicine, okay, where we use pharmacogenetics. That's a study of drugs and genetics together. Pharmaco is drugs, and genetics obviously is our genome, and we can then make a good choice in terms of the personalized drug that we give to that person, and it will be at the most effective. This is the key word. At the most effective dosage, as well as choosing the most effective drug for that person. And that is an example of personalized medicine. I think for all you young people that are studying the higher course just now, you will probably experience this in your lifetime, where you will be getting not just the generic antibiotics for a chest infection, but you'll be getting a specific drug at the particular dosage that is going to best treat your chest infection. Okay, so they'll be looking at your personal genome sequence to help them with that. And this study of drugs and genetics and the relationship between them is called pharmacogenetics, and it helps in terms of the choice of drugs for that particular person. We can also look at the person's genome as well to analyze it to predict the likelihood of developing certain diseases. Okay, so that's the human genomics topic, and that is the main things that you guys need to know about for your exam. Now, as I say, it's not used in great depth in the exam in previous years. That's not to say it won't come up this year, and you have to know all of this stuff. But I'm going to show you a couple of questions that have come up in the last few years in the multiple choice section, and obviously to go about answering them. So I hope the lesson part's been useful, a nice recap over those parts of the human genomics topic. Let me know if there's anything you struggled with or you still have questions about. Hello and welcome to Lesson 6 of Unit 1 of the Higher Human Biology course. This lesson is going to be focusing on Topic 6, which is metabolic pathways. So this is a tricky topic. It's one that students often find is one of the most difficult when it comes to the exam. It's also quite heavily examined, so you tend to find a question on it every year in the exam, and it's usually an applied context type question where they try and apply it to a new or unfamiliar context for you. So these can be quite tricky ones. So we'll need to make sure that we go over things thoroughly on this topic and that you get a good understanding of it by the time that we've finished and you've gone over and did a bit more of your own studying as well. So what are metabolic pathways? Well, metabolic pathways are a series of reactions which happen within cells within the body. And those reactions can be involved in many of the major processes that our cells undertake, such as aerobic respiration and protein synthesis that we have learned about so far. Metabolic pathways they're integrated and controlled pathways of enzyme catalyzed reactions within the cell. So they're integrated. That means that all the steps happen within the cell. They're controlled by enzymes. Okay, they are enzyme catalyzed reactions. Now, when we were at National 5 Biology, we learned about the likes of respiration and photosynthesis as enzyme controlled reactions. And these are a good example of some metabolic pathways that we already know about. Okay, so they're controlled by enzymes, which means that the concentration of enzyme will have a big impact on how fast these reactions go, because as we know, enzymes do speed up these reactions. Now, metabolic pathways can have reversible steps, irreversible steps, and alternative routes. And if we have a wee look at our diagram here, we can see some examples of those. So this here, the substrate has been broken down into a molecule. We'll just call it intermediate 1 for the sake of this. Now, that is an example of an irreversible reaction because the arrow is only going in one direction. The breakdown of intermediate 1 to intermediate 2, it does go this way, but it also goes back the other way as well. So we call this an reversible reaction, okay? It's a reversible reaction, so it can go both ways. If we have a high concentration of intermediate 2, for example, we can convert some of that back to intermediate 1. Okay, so that's reversible. And we also have this alternative route as well. Now, that will probably only come into play really if, for example, let's say this enzyme here, for whatever reason, stops working. Okay, and that will cut off this part of the pathway. The substrate, in order to get to the product, will have to go via an alternative route. Okay, and there's usually another intermediate product here that the substrate will break down into, and then that intermediate product will break down into the end product. Okay, and we see that in the likes of aerobic respiration, amongst other things. Now, reactions within the cell can either be anabolic or catabolic. Now, at National 5, we learned about synthesis and we learned about degradation. Now, anabolic and catabolic reactions are very similar to our synthesis and degradation reactions we already know about. And we can see just at the bottom of the screen here, we've got our anabolic and our catabolic reactions here. And we can see that the anabolic reaction here is building up from larger molecules, two larger molecules from smaller molecules. So we've got our small molecules here. We are putting in energy. This requires energy. And we're getting our larger molecules here, okay, from small to large requiring energy. Over this side here, we've got the catabolic reactions taking place. That involves the breakdown of our large molecules into smaller molecules, and this reaction releases energy. Okay, we can see that there, large molecule all chained together. It is broken down, it releases energy, and we get all these small molecules as products at the end. Okay, so you need to remember which of these reactions is which. If you think about anabolic a bit like the synthesis reactions we learned about at Nat 5, and catabolic like the degradation reactions that we learned at National 5 as well. Okay, so hopefully all of that makes sense. Metabolic pathways controlled by enzymes. We have reversible, irreversible, and alternative routes, and our reactions can either be anabolic or catabolic, and we need to know the difference between those two for our exam. Okay, so metabolic pathways are controlled by the presence or absence of particular enzymes and the regulation of the rate of reaction of key enzymes. So what does that mean? Well, if the enzyme is present, the reaction and the pathway are therefore going to proceed and they're going to go faster. If the enzyme is absent, then obviously that pathway will come to a halt. And if we want to, for example, slow down a reaction, so people often talk about our metabolism. Now, our metabolism is controlled by the sum of all of these reactions that happen in our cell. So the more of these reactions that are happening, the greater our metabolism, or the higher our metabolism. The less of these reactions that are taking place, the lower our metabolism. And that is all controlled by these enzymes. If they're in higher quantities, our metabolism is going to be greater. If they're in lower quantities, our metabolism is going to be lower. Okay. So how do enzymes actually carry out their reactions? So a quick recap on Nat 5. Now, we learned at Nat 5 that on the enzyme we have an area called the active site, and that active site binds to a specific substrate. Okay, and that's really as far as we went at National 5. The shape of that active site was complementary to our specific substrate, wasn't it? Now, in an ideal world, that would be perfect and it would be tie that in a neat little bow, and that's how we'd understand it, but it's not quite how it works. Now, when the substrate does enter the active site, the substrate doesn't have quite the same shape, okay? The shape is just slightly different. Okay, it's very similar, but it's just slightly different. Now, what needs to happen then is for the active site actually needs to change shape, okay? The active site changes shape. Okay, and the reason it does this is to better fit the substrate after the substrate binds. And this process is called induced fit. Okay, and we can see it in the animation over here on the right. The substrate enters the active site, but when it initially enters, the active site isn't quite snug on that particular substrate, and therefore it has to change shape to better fit the substrate. Now, induced fit is good for reactions because it lowers the activation energy. That's the amount of energy that is required for the reaction to get started. Okay, and obviously with a lower activation energy, that's better for cells because they can start the reaction sooner and by using up less energy. And that's going to be good in terms of speeding up our metabolic pathways. Okay, so induced fit lowers that activation energy required for the reaction as the substrate is more closely bound to the active site. Now, the substrate molecule has a high affinity for our active site. That means it has a high level of attractiveness for the active site. It wants to bind to it. Okay, so two molecules with a high affinity want to bind with each other. All right. Now, once the reaction has taken place and we have our products, those products leave the active site because they have a low affinity for it. Okay, products have a low affinity for the active site, which is why they are then able to leave. That's why when the substrate goes in, that molecule can eventually leave, because when the reaction's taking place, the products have a lower affinity. Okay, so affinity, that's the attractiveness between two molecules, and the substrate has a high affinity for the active site. The product has a low affinity for the active site. Quick shout out, as always, to the Amoeba Sisters. They've got a nice little video that shows nice animations of how enzymes function and how induced fit functions. So if you want to see that, you can go and subscribe to their channel and watch the video. Okay, so what other factors can affect the rate of enzyme reaction? How fast that enzyme goes, the enzyme activity, to use the term that we used in National 5? Well, they can be affected by a few things. The first thing we're going to have a look at is something called the substrate concentration. And simply put, that is the concentration of the substrate molecule that binds to the active site. If we have lots of that substrate, we're going to have more enzymes binding to the substrate and therefore more enzyme activity. The rate of the reaction will increase. And we can see that in our little graph here. We have the enzyme activity increasing here, and the graph continues to go up as the substrate concentration also increases. However, we reach a point round about here on the graph where the enzyme activity starts to level off. Okay. Now, at this point, continuing to add substrate actually makes no difference to the reaction rate. And the reason for that is because all of the active sites that are on our enzyme are occupied by substrate. Okay. So it's like 20 students walking into a classroom and there are 20 seats. Okay. Now, initially, pupils will be able to find a seat and those seats will fill up and fill up and fill up, and we'll have more and more students seated. But then if we continue to just keep adding students to the class, once those 20 seats are occupied, it doesn't matter if we keep adding students to the class, they won't be able to have a seat and they won't be able to sit down for the lesson. Okay. And it's a bit like that in terms of enzymes and substrate. We can keep adding substrate all day long, but because all the active sites are occupied, it will make no difference to enzyme activity. Now, some metabolic reactions are reversible. We spoke about this earlier on, okay, that word reversible, which means that the product can be reverted back to the substrate, okay, we can reverse that reaction. And the presence of a substrate or the removal of a product can drive a sequence of reactions in a particular direction. So, for example, in something like aerobic respiration, the presence of glucose will drive aerobic respiration. Okay, it will drive that series of reactions that occur to produce our ATP at the end. And similarly, if you remove a product, that may well stop that metabolic pathway, and we'll have an accumulation or a buildup of a particular intermediate substance in the middle of the pathway, unless it's reversible, of course, in which case it can go back to the initial substrate. Now, inhibition is the last part of this topic, and there are three main types of inhibition you need to know about for your exam. The first of which is competitive inhibition. Now, competitive inhibitors are inhibitors which bind at the active site, okay, and by doing so, prevent the substrate from binding. So if we have a look here at a normal reaction, we've got the enzyme here, we've got the substrate here, and the substrate's able to enter the active site, and we get an enzyme substrate complex. Okay, all good. But when this red inhibitor here is present, there is now a 50-50 chance that that inhibitor will reach the active site before the substrate does. In which case, if it does, it will block the active site and prevent the substrate from binding, which means there's going to be no reaction on that enzyme. The way that we overcome this, the way that we get around this competitive inhibition, the way that we reverse it, is by increasing the substrate concentration. And if we look at our diagram to understand that, it's, for example, we keep the inhibitor just at 1 there, but we make the substrate 4, okay? So we've increased the substrate concentration. That now means that instead of being a 50-50 chance that the substrate will bind, there's now an 80% chance that the substrate will bind first, okay? The inhibitor's got a 1 in 5 or a 20% chance of binding, but the substrate has got an 80% chance. So therefore, if we keep adding substrate, we're going to eventually overcome the inhibitor, okay, because we're lowering and lowering the inhibitor's chance of getting there before the substrate. So the reaction rate, the enzyme activity, will continue to increase. The second type of inhibitor we need to know about are non-competitive inhibitors. Now, those ones are slightly different because they bind away from the active site, not on the active site. But when they do that, when they bind away from the active site, as we can see in the diagram here, the non-competitive inhibitor changes the shape of the active site, so it's a kind of square shape here initially, and it changes it to this sort of V shape. And what that means is the substrate is not directly competing with the inhibitor, but because of the inhibitor's action, the substrate can no longer bind to the active site because the shape has been changed, and therefore the substrate can't bind to it anymore. So a non-competitive inhibitor binds away from the active site, and it changes the shape of the active site, which prevents the substrate from binding. Now, a non-competitive inhibitor cannot be reversed by increasing substrate concentration. And we can see from our graph here, the non-competitive inhibitor, no matter how much substrate we add to it, the non-competitive inhibitor does not increase enzyme activity there, okay? The graph just stays level, so it remains low throughout, okay? And we can compare that to the competitive inhibitor, which eventually, with added substrate, will overcome inhibition, and compare that to the initial no inhibitor graph, where we see the enzyme activity increasing until all the active sites are occupied there. And lastly, feedback inhibition. Now, feedback inhibition is a little bit different. Feedback inhibition is to do with the end product. Now, if we go back to the pathway that we had earlier on, this metabolic pathway here, we can see a substrate being broken down to an intermediate, another intermediate, and then the end product. And there are three enzymes that help us to catalyze those reactions. So feedback inhibition is when this end product, okay, that's this thing here, the end product, in the metabolic pathway reaches what we call a critical concentration, okay? Essentially, at the point where we don't need to make it anymore, because it starts becoming a waste of the cell's resources and energy to keep making it because the cell doesn't require it. So when it reaches that critical concentration, the end product itself will then move back and it will find an earlier enzyme, okay, in this case, enzyme 1, and it will inhibit that enzyme, okay, so it inhibits that earlier enzyme. And what that means is that it will block that pathway, okay, so that the substrate can no longer be broken down, and therefore the reaction and the metabolic pathway will not continue, okay. It prevents further synthesis of the end product, okay. This will not be produced because none of the pathway will proceed, and the substrate is no longer able to be broken down into the intermediate because this enzyme here, the first enzyme, is being inhibited by the end product. Okay, so we call that feedback inhibition because the end product is going backwards in the pathway and inhibiting that first enzyme. Okay, we're going to stop the lesson part there, and we're going to go and have a look at the past paper questions as we always do. I hope that was useful for you as a recap. Hello, and welcome back to lesson 7 of the unit 1 of the higher human biology course. Topic 7 is all about cellular respiration. So we're going to be building upon the knowledge that you learned at National 5 on respiration, and obviously going a bit deeper as we always do at higher. So cellular respiration is going to be split into 3 different stages. The first stage that we're going to focus on is glycolysis, and glycolysis takes place in the cytoplasm of the cell. Now, we learned that glucose is broken down into 2-pyruvate and 2P back in National 5. And what we're going to be doing today is we're going to be looking at why that is, what happens in between to enable us to get those 2-pyruvate and those 2P. And we'll look at all the stages involved. So glycolysis, generally speaking, the overall reaction is the breakdown of glucose to pyruvate in the cytoplasm. That's the summary of the process. But as you can see from looking at the diagram, it's a lot more detailed than that. Now, it's a process which actually requires us to put energy in order to get those ATP out at the end. So it's split into 2 different sections here. We have the energy investment phase, okay, the energy investment phase. And during that energy investment phase, we have to put 2 ATP in. And we do that both here and here in the energy investment phase. Now, the reason that we put those ATP in is to give us the energy to carry out something called phosphorylation of glucose into the intermediate 1. So the phosphorylation of glucose requires this ATP here, and the phosphorylation of this intermediate molecule also requires that ATP. So to allow this metabolic pathway to continue, A couple of these reactions require require the ATP for phosphorylation of these molecules. So I'll say that one more time. In order for the metabolic pathway to continue and for glucose to be broken down into pyruvate at the end, a couple of these reactions require ATP for the phosphorylation, first of all, of glucose and then of the intermediate. Now, all of this stage here, from here down to here, is known as the energy investment phase because we are putting 2 ATP in to this process. Don't worry too much about the NADH and NAD for now. We'll talk a little bit more about them in the next stage, but just note that this reaction occurs here as well. Okay, but we'll come back to it a bit later on. So at the end of the energy investment phase, we have put 2 ATP in for these 2 phosphorylation reactions to happen. Then we move on to the energy payoff stage. This is where we start getting the ATP back out. And in total, in the energy payoff stage, we get 4 ATP back out. The energy payoff stage results in what we call a net gain of ATP. Those of you who study business and so on will know all about net profits, etc. It's the equivalent, but for biology. So we end up with more ATP because 4 ATP are being produced here and 2 ATP went in, and that gives us our net gain of 2 ATP that we will remember from that 5 was the yield of ATP in this process. And we can see those 2 processes here, which produce those 2 and 2, in total 4 ATP. And if we remember as well from National 5 that it's the addition of a phosphate to ADP that builds up the ATP molecule, and vice versa. The breakdown of ATP produces ADP and a phosphate. And it's that phosphate actually that's used in the phosphorylation, okay? Basically that phosphate gets added to the glucose and becomes this molecule here. And similarly, that phosphate there gets added. That's what phosphorylation is to add the phosphate, the addition of a phosphate molecule. And then lastly, we're in aerobic conditions. And the rest of this topic is going to assume that we are. That means we have oxygen. If that happens, pyruvate will be further broken down. This pyruvate here is going to be further broken down into another molecule. And that molecule is known as an acetyl group. Now, we'll talk more about that in the next slide. But that acetyl group is going to combine with something called coenzyme A to form something called acetyl coenzyme A. But we'll see that in the next diagram here. So we've got that pyruvate that was produced at the end of glycolysis, and it combines with coenzyme A. These two combine together here, and they produce acetyl-CoA when we have aerobic conditions. Now, that allows us to then move on to this stage here, this circular stage here, which is known as the citric acid cycle. Citric acid cycle. Now, that occurs in an area of the mitochondria known as the matrix. We know that the second part of aerobic respiration from National 5 takes place in the mitochondria. We're now going to be more specific about where and about what happens within the mitochondria. So the citric acid cycle occurs in the matrix of the mitochondria. That is very much well worth memorizing because it does come up in the exam very regularly. And another thing just to add on that note, you should always say the matrix of the mitochondria, not just the matrix, because there are many matrices within the human body. So the matrix of the mitochondria is where this occurs. So in the citric acid cycle, the acetyl group from acetyl coenzyme A combines with the oxaloacetate here to form a molecule known as citrate, which is where the citric acid gets its name from, the citric acid cycle, because we're eventually going to regenerate continuously that citrate molecule there. Okay, so acetyl-CoA combines with the oxaloacetate, forms citrate. We can see that happening here. Acetyl-CoA, oxaloacetate forms the citrate. Now, the rest of the cycle, when I was a student at school, I had to learn all of the intermediate molecules here that citrate was broken down into before it became oxaloacetate again. However, you guys are lucky, you do not need to know that. All you have to say is that a series of enzyme-controlled steps takes place. These are the enzyme-controlled steps all the way around the rest of the cycle here. You don't necessarily need to know the specific molecules, the intermediate molecules here. Now, eventually, citrate is going to be gradually converted back into oxaloacetate, okay? Going around this cycle here, it's converted back into oxaloacetate. And during that series of enzyme-controlled steps, there will be the generation of a little bit more ATP. We can see that here. There will be the release of carbon dioxide. And again, we can see that here in our citric acid cycle. So a couple of products here from all these series of reactions that are happening. Now, in addition to that, there is a set of enzymes called dehydrogenase enzymes. Now, if we break down that word, dehydrogenase enzymes, we know it's A's, which makes it an enzyme, and it works with hydrogen. It's got a role in removing hydrogen here. Now, what it does is it removes both hydrogen ions and electrons from the molecules in the citric acid cycle, and it then passes them onto a coenzyme called NAD, and when that combines with the hydrogen ions and electrons, it forms another molecule called NADH, and we can see that happening over here. So the dehydrogenase enzymes will be working right in this area here to remove hydrogen and electrons and then combine those with the NAD to form NADH. Now, this reaction with the dehydrogenase removing the hydrogen ions and electrons, that occurs in both glycolysis and the citric acid cycle. So we see it here in the citric acid cycle, but we also see it back up here in glycolysis in this region here. So that reaction happens in both steps, in both glycolysis and the citric acid cycle. And I'm just emphasizing that because that has been asked in a past paper question before, and it's well worth remembering that. Students often remember it happening here in the citric acid cycle, but forget that it also happens during glycolysis. And then finally, those hydrogen ions and electrons that are part of the NADH are passed on to the next stage of the process, which is known as the electron transport chain, and we'll get to that in a second. And that's located on the inner mitochondrial membrane. So let's look into that. The electron transport chain and ATP synthesis. So the electron transport chain is a series of carrier proteins, okay? That's these things here: carrier protein, carrier protein, carrier protein, protein. We can see that series of carrier proteins there, 1, 2, 3, 4 in the diagram. Now, they're attached to an area called the inner mitochondrial membrane, or the IMM, as I think of it in my head for short. Now, remember, that's a different part of the mitochondria from where the citric acid cycle takes place. That takes place in the matrix. This stage, the electron transport chain, is in the IM, or the inner mitochondrial membrane. That's this area here. Now, the electrons that were brought by the NADH, they brought along hydrogen ions and electrons. The electrons are passed along the electron transport chain, releasing energy. So those electrons, we can follow their path there. There's one electron, there's another one, and another one, and then eventually they leave. So they'll pass all the way along this electron transport chain, this series of carrier proteins, and they will release energy as they do that. Now, why do they need to release energy? Well, that energy is going to allow hydrogen ions, the same ones that the NAD transported, the hydrogen ions to be pumped across the inner mitochondrial membrane. And you can see the hydrogen ions getting pumped through each of these proteins here. The hydrogen ions get pumped across the membrane. Now, the reason they have to be pumped across the membrane is because if we look at the diagram, the concentration of hydrogen ions on this side of the membrane is much higher than this side of the membrane. So they're traveling by active transport. They're going against the gradient, which is why we need the energy from the electrons to actively pump the hydrogen ions across the inner mitochondrial membrane. So I hope you're following me so far. Finally, because we have this high concentration of hydrogen ions on this side, they are going to be able to flow. Okay, they're going to be able to flow, and it's important you use the correct terminology here. They're going to be able to flow back through this protein here, which is called ATP synthase. Okay, ATP synthase, this membrane protein. And as the name suggests, synthase, it's an enzyme, and as its name suggests, it synthesizes or produces ATP. And we can see that reaction happening there, where ATP is generated from ADP and phosphate. So the flow of hydrogen ions through ATP synthase generates ATP. All right. So I think that's the trickiest part. The last wee part is what happens to all of the things now that the ATP has been produced. Well, the hydrogen ions that are flowing through here, they combine with the oxygen that we needed to be present for aerobic respiration, and the electrons, which are now low energy electrons, And they combine together, all three of those molecules, hydrogen, oxygen, and the electron, and they combine together to form the molecule water. They form water. And that makes sense. Hydrogen and oxygen, H2O, that forms water. But don't forget that the electrons also combine with the oxygen. Hydrogen and the electrons plus water plus oxygen equals the water. And the ATP that's used is, and the ATP that's generated here is used to transfer energy to cellular processes, okay, or cellular processes which require energy. Now, at National 5, we learned about those kind of cellular processes. They were things like muscle contraction, protein synthesis, cell division, and the transmission of nerve impulses. But there's way more than just those. Okay, so all of these processes require ATP, and the ATP generated by the electron transport chain is used to help us carry out those other processes and breathe. So that's a long and complicated little process. So I'll just do a quick recap before we move on. There are three steps, three main stages. We have glycolysis, which takes place in the cytoplasm, and glycolysis is the breakdown of glucose into pyruvate. We'll go back to that one. We have an energy investment phase and an energy payoff stage, which results in a net gain of ATP. The ATP that we put in is used for the phosphorylation of glucose and the intermediates. And when we have aerobic conditions, the acetyl group will combine with the pyruvate. The pyruvate will be broken down into the acetyl group and combine with the coenzyme A to form acetyl coenzyme A. We then have the citric acid cycle, which occurs in the matrix of the mitochondria. During the citric acid cycle, that acetyl group from the acetyl coenzyme A will combine with the oxaloacetate to form citrate. There'll be a series of enzyme-controlled steps where citrate is gradually converted back to oxaloacetate, and we also get the net products of ATP and the release of some carbon dioxide. In addition to that, dehydrogenase enzymes will be removing hydrogen ions and electrons during both glycolysis and the citric acid cycle, and they'll pass those on to the coenzyme NAD, which forms NADH. The hydrogen ions and electrons from the NADH are then passed on to the electron transport chain, which is located on the inner mitochondrial membrane, and it involves these series of carrier proteins that the electrons will then be passed along those proteins, and they will release energy as they go. This energy allows the hydrogen ions to then be pumped across the inner mitochondrial membrane, and the flow of those hydrogen ions back through the membrane protein ATP synthase, results in the production of a large quantity of ATP. Finally, all of those things that are left over, the hydrogen ions, the electrons, and the oxygen that we've breathed in for the aerobic conditions, they will all combine together to form water. And the ATP, the large number of ATP produced by the electron transport chain, can then be used for cellular processes like muscle contraction, cell division, protein synthesis, transmission of nerve impulses, and many, many others. Folks, so that was quick. We went through quite a lot there. There's a lot to study. I would suggest with this topic, you're going to have to sit and this is one that would be good to start drawing some diagrams, sketch out the glycolysis, the citric acid cycle, and the electron transport chain. Maybe by having a look at your notes first, then covering those up, having a draw at it, see how much of it you can recall, looking back, drawing it again, and so on. Because there are quite a few diagram questions that come up in the exam relating to these. We also get a lot of questions in the exam which are extended response questions, so the longer answer questions. And again, they can be on one or two of these steps in the cellular respiration process. It's rare for you to get asked about all three, but it can be on one or two of these steps. So it's a good idea to memorize as much of the processes and the reactions that happen throughout cellular respiration in case you do get a question on that. Hello, folks. Welcome to lesson number 8 of Unit 1 of the Higher Human Biology course. Lesson 8, or topic 8, is all about energy systems and muscle cells. Okay, so we're going to be looking at lactic acid metabolism, and we're going to be looking at slow and fast twitch muscle fibers as well. So you'll probably look at this diagram on the slide here and think that looks a little bit familiar. And you'd be right in that the first part of the diagram is glycolysis from the previous topic. And on the right-hand side of the diagram is something we learned about in National 5, which was fermentation in animal cells. So we're going to look at how that works at higher level. Obviously, there's a few more details than what we've previously learned, and hopefully you'll understand this topic a little bit better afterwards. I should say this topic often goes hand in hand with cellular respiration in the exam papers. We don't often get separate questions on this topic, but often they integrate it alongside the respiration question because based on the content, you can see that they sit well alongside each other. So what is lactic acid metabolism? Well, during vigorous exercise, the muscle cells do not get sufficient oxygen to support the electron transport chain. Okay, so you're doing the 400 meters or the 800 meters or something similar, and you're going pretty much full out for quite a long time. Over the course of that exercise, your cells will not get enough oxygen. And if they don't get enough oxygen, that means they can't support the electron transport chain, which means you will not generate the bulk, the majority of your ATP. Now, we know that we do have the ability to generate 2 ATP from the breakdown of glucose to pyruvate, and we know that that does not require oxygen. So we can still produce some ATP, just a very small quantity, only 2 ATP, compared to the large numbers we can create via the electron transport chain. Now, under these conditions where we lack that oxygen, the glucose is broken down into pyruvate, but then the pyruvate is converted to something called lactate. Now, this conversion involves the transfer of a hydrogen, okay? So a hydrogen needs to be added to the pyruvate to form the lactate, and that hydrogen comes from our coenzyme from the last topic, NAD. Okay, NAD is transporting the NAD in the form of NADH, and it is then releasing that hydrogen to combine and form the lactate. Now, this is useful for us because it regenerates the NAD, which can then go back to glycolysis and pick up the hydrogen that the dehydrogenase enzymes have managed to remove. Okay, so it's a nice cyclical process here, and we need that because we need glycolysis to keep happening, to keep producing those 2 ATP, because this is our only source of ATP at the moment. So we need that constant regeneration of NAD. Okay, otherwise we just wouldn't have any ATP. Now, the issue with this method of generating ATP is that over time, lactate accumulates. This lactate here builds up within the cell. So as the lactate accumulates, that will cause muscle fatigue and the buildup of an oxygen debt. Now, an oxygen debt is essentially the oxygen that your body requires for the exercise that you're doing, but you're not actually getting into your body. Okay, so if you imagine the amount of oxygen you would require to generate ATP to run 400 meters, you're not able to get that into your body during the 400 meter race. So we call that your oxygen debt, i.e. how much oxygen you will then need to get back into your body after the race. Okay, that's when you repay your oxygen debt. And what that will allow you to do, all of that oxygen, is to allow respiration, aerobic respiration, to provide all that energy, all the additional ATP, to convert the lactate, convert this lactate back into pyruvate and into glucose within the liver. Okay, so that's why we need to repay that oxygen debt at the end of the race, to allow respiration to provide the energy to convert lactate back to pyruvate and glucose in the liver. Okay, so just one more run through there. So during vigorous exercise, the cells do not get enough oxygen to support the electron transport chain. Under these conditions, the pyruvate down here is converted into lactate. Okay, pyruvate to lactate, pyruvate to lactate up here. The conversion involves the transfer of hydrogen, which comes from this molecule here, NADH. Okay, the NADH, that transfers the hydrogen here. That hydrogen allows lactate to form. That NADH comes from glycolysis. That NADH initially came from glycolysis. We'll remember that from the respiration topic. And the regeneration of this NAD, this molecule here, then is needed to allow ATP production to happen during glycolysis. So this coming back here allows this pathway to proceed, and that generates, as we know, our ATP later on. And because we're not undergoing aerobic respiration, we need all the ATP we can get. So we're grateful for those 2 ATP that glycolysis can provide us. So we need the NAD to be regenerated for that purpose. But the problem is, over time, lactate accumulates and muscle fatigue occurs, and we also build up our oxygen debt. Now, we can repay that oxygen debt when the exercise is complete. That's why you can't rely on this non-aerobic respiration for too long, because eventually you've got to start breaking down that lactate. And what this does, when we repay the oxygen debt is it allows aerobic respiration to occur, which provides us with loads of ATP, and that provides the energy to convert the lactate back. You can see this reverse arrow here, converts it back into pyruvate, and also the pyruvate can be reversed into glucose if needed in the liver. Okay, so hopefully lactic acid metabolism makes sense there. If not, have a few looks over that. Again, this is one of those that might be useful to sit and draw the diagram just to sort of visualize it and get some muscle memory going in terms of knowing that process. So moving on to the types of skeletal muscle fibers. Now we're going to be looking at two types. We're going to be looking at slow twitch and fast twitch muscle fibers. And in some ways, this is a straightforward topic because actually slow twitch and fast twitch muscle fibers tend to be the opposite in almost every way. Okay, and so when we're describing slow twitch, for example, fast twitch is, relatively the opposite and for most of the characteristics. So we'll have a look at them both just now, and hopefully we can distinguish the difference between them by the end of this lesson. So slow twitch muscle fibers contract relatively slowly, but they can sustain their contractions for much longer. Okay. Now this here, when it says contracts relatively slowly, someone like Mo Farah with all these slow twitch muscle fibers is still moving pretty fast. It's just it's contracting relatively slowly compared to the fast twitch ones. Okay. So it's not actually a slow contraction. It's just relatively slowly. And that's worth remembering when you're writing your answer in the exam. But the good thing about the slow twitch muscle fibers is they can sustain contractions for longer. What that means then is that they are useful for endurance activities, things such as long distance running, cycling, or cross-country skiing. And we can see a couple of examples of some athletes here in both of those disciplines. Now, slow twitch muscle fibers rely on aerobic respiration. Okay. Why? Because they need to generate a lot of ATP. They need a large number of ATP for these endurance activities. So if you need to generate a lot of ATP, you also need to have many mitochondria. Okay. We also need a good blood supply. Why do we need a large blood supply? It's to provide us with the glucose and oxygen that are carried in the blood and are needed for aerobic respiration. And we need a high concentration of an oxygen storing molecule, which is called myoglobin. And this is a protein that's found within the muscle cells. And it's the oxygen storing protein that allows you to, for example, hold your breath underwater for a while. Okay. The reason why your cells still have access to some oxygen even when you're holding your breath is because of this molecule here. So a high concentration of that is useful because at times you'll be moving so fast in your endurance event or working your muscles so hard that you might not just breathe in enough oxygen. So having some stored in the myoglobin will be of great benefit. Their major storage fuel is fats for slow twitch muscle fibers. Now, fast twitch muscle fibers, as I said before, are in many ways the opposite of slow twitch because they contract relatively quickly. Okay. And again, it's all relative. Both of these muscle fibers are actually contracting reasonably quickly, but it's just that the fast twitch ones contract even faster relatively quickly compared to the slow twitch fibers. And they do it over shorter periods. Okay. So they're not contracting for as long. Now, they're useful for activities such as sprinting and weightlifting, which we can see in our two images here. Okay. And fast twitch muscle fibers can generate ATP through glycolysis only. Now, as we know, glycolysis only produces P through that process. So therefore, these have to be fast activities. These need to be powerful activities, okay? Because we can't sustain them for very long periods of time. So we use glycolysis only to generate the ATP that we need for this. As a result, because we don't have much aerobic respiration happening in these cells, we have fewer mitochondria and we have a lower blood supply because we don't need to provide the oxygen for aerobic respiration here because we've got glycolysis only. So no oxygen needed. So the blood supply, it can be a little bit lower compared to the slow twitch muscle fibers. And notice how it's always fast twitch can blah, blah, blah compared to slow twitch muscle fibers. Okay. Similarly, it contracts relatively quickly. And that again is relative to slow twitch. So we're always comparing the two. And the questions that we get in the exam tend to be comparison questions. You know, what fast twitch is this, what is slow twitch, or et cetera, and you're always comparing them to each other. Now, the major storage fuel of fast twitch muscle fibers is glycogen. Now, as we know, glycogen gets broken down into glucose, which we need for the glycolysis reaction that happens in these fast twitch muscle fibers. So that's why the major storage fuel for fast twitch muscle fibers is glycogen. Okay, so we have a wide range of these muscle fibers within different muscles within our body. Now, most human muscle tissue contains a mixture of both the slow and fast twitch muscle fibers. Athletes tend to show a distinct pattern of muscle fibers that reflect their sporting activities. So, for example, we saw Mo Farah in the previous slide there. It is likely that if we did a cross section of his muscle fibers, it would either look like here or like here, okay? Either the long distance or middle distance runner, okay? We know that Mo Farah actually, for a middle distance runner, is very fast. He's got a very fast sprint, so he probably has more in the middle here, where he has a mixture of fast and slow twitch muscle fibers. Whereas someone like Usain Bolt, who was a very fast sprinter, he probably, if he was to run middle distance like 5,000 meters, 5 kilometers, he would probably struggle with that, because I would imagine if we did a cross section of his muscle fibers, we'd see something like this one on the right here, where he has lots and lots of fast twitch muscle fibers, but very few white ones. So if we looked at people who are sort of elite in different types of activities, and we put them into endurance or like sprint activities, we would see this distinct pattern within their muscle fibers. And a little summary table there. It might be worth you guys writing down a summary table for this topic, because what it does is it allows you to very quickly see the difference between the slow twitch and the fast twitch muscle fibers, and to see how they're different to each other in all of these different features. Okay, so this is probably worth having a wee look at, pausing the video and maybe writing this down if you don't already have a copy of it in your notes.